Hi, this is Mr. Waite, and I'm going to attempt to show you how to do the mitosis and meiosis virtual lab. I'm going to go and let you read all the instructions on here. Highlight a few important things are that you will need to access SharePoint, and you will need to pull up the mitosis virtual microscope file. You will need to be able to identify the different stages of mitosis as well. And then one important thing on here is you will be counting a total of at least 100 cells. So if I go back to my SharePoint site right here, let me go back to where we start from. Here's the AP Biology. Scroll down here to cell reproduction, DNA replication. These are our different stages of mitosis, including interphase over here. And if you don't feel comfortable with those, please click on this link right here, and that'll help you identify those different stages. But I'm going to open up the mitosis virtual image file. I'm going to go into here, and I need to count 100 cells of the onion root tip cells. So I'm going to try to find an image that I think is easy to identify. And I kind of like that one. Let's see what other ones there are on here. And let's go ahead and let's do this one right here. All right, so you could start with any of these. You might find all 100 cells on one slide, but you might need to refer to the other slides as well. So I'm going to use this one. By the way, the lab calls these frames. So if I go right here, I'd like to identify a cell that's an interphase. Interphase has the nuclear membrane intact. It has the chromatin condensed right here. So this particular cell I'm going to identify as interphase. I for interphase. If you print these off, the images, they're easy to write on, just like I'm doing right now too. Here's another one that's in interphase. Let's find one that's in prophase, where the chromosome is actually condensing. And so this looks like a good one for this particular cell. I would call this one prophase because I can see the chromosomes. It doesn't look like this chromatin that's the spaghetti structure that's tightly wound up right here. I'm actually starting to see some of the chromosomes over here, again in prophase. Let me make that just a little bit wider. Prophase, next phase is metaphase, where they meet in the middle. And I might go ahead and count this one as being metaphase. And you can see if I drew a line right here, it looks like these chromosomes are meeting in the middle. Anaphase is where they are being pulled apart. So I find this cell over here, and I'd call this cell anaphase. You can see the chromosomes are going one way and the other. And then telophase is where they're actually all the way apart, and they're starting to form a new cell, two new cells actually. So let me see if I could find a good example on here. It's the most rare one, so it might be hard to find if I can find an example at all. In fact, I would say, I know it looks like two different cells right here. If this could be an example of telophase, where they've separated and we're starting to see the cell plate form right here. So I would call this one telophase, but really that's the only one that I see. You should see most of the cells in their interphase stage, followed by prophase, metaphase, anaphase, and telophase in that order. So if I go back here to the virtual lab, I'm going to want to record these. Please don't use the same numbers that I'm doing. I'm just going to make up some numbers right now by going back here. And I'm going to say, you could just make tally marks here. These are supposed to be the different fields of view that you're looking at. Again, if you get all 100 in one slide, you can just write them here. If you need to count another slide, go ahead and count them over here too. I'm going to say for my interphase, I found 60 total. Let's change the lead. And for my prophase, I found 20. Interphase 10, anaphase 7, and telophase 3 out of all the 100 cells that I looked at. So the next thing it says over here is it says the percent total cells counted. Because we already did 100, 60 out of 100 would be 60%, 20%, and so forth. That's easy. The time in each phase, if you go back and read the top of this lab, it tells you that this was a total of 1,440 minutes. And so what I'm going to do is go back to my previous slide where I had it back here. And if I said that this one was 60, and I know that the total time was 1,440 minutes, Then I'm going to say, well, I'd like to take that times 60% or by 0.6. And what I do, if I do that, let me just grab my calculator here really quickly and do that. I would say 1,440 times 0.6, I get 864. So over here, the total time spent in this phase, according to the picture, would be 864 minutes. Over here on this side, it's asking you to make a pie chart. So if I'm supposed to represent 60%, I might come over here and say, okay, this is about 60% right here, and this 60% of the time is spent in interphase. Obviously, I'd want to make sure I get the other phases on here for my pie chart as well. And once you do that for the cells, for the onion root tip cells, you're going to want to do the same thing for the animal or fish, whitefish blastula cells. And those were back on the virtual lab. If I go back there, if I scroll down a little bit more, these are onion root tips. If I go down a little bit more, still some onion root tips, and go down even further, here's our whitefish cells. Those are hard to see, but I've given you a few different slides so that, again, you need to count a total of 100 cells. And hopefully you can find those cells. I know that's not easy to see. Do the same process. Now let's go back to the lab and look at the other side, which is the, actually go ahead and answer these questions right here for the mitosis analysis. And this says conclusion for assignment 1. One thing that a lot of students missed last year is make sure that you make an inference based on each stage of cell division about the relative length of time. Don't just list one stage, list them all. Down here, make sure you answer that. And then let's talk about part 2, meiosis and crossing over in Sordaria. So let me explain this really quickly. When you go back to my website, and let me go back here, I do ask you in the directions to watch this video first. This is the Sordaria lab video description. Mr. Anderson will give you a complete description of what's going on first. Once you get there and you're ready to start counting, then you can open up the meiosis file and find those pictures. There's a few frames here that show you what the crossing over looks like, and then it starts to show you some actual pictures. These are some of the pictures right here, indicated by number 1. So here's an example of crossing over. And for this one, what I see right here is this one is tan, tan, black, black, tan, tan, black, black. If you can see that, let's make this a little bit thicker. Let's do another one. This one up here would be tan, tan, black, 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 tan, tan. And this one over here, kind of the opposite thing. Black, black, tan, 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 black, black. Now if you go back to the lab, they're going to ask you to record all the different number that you see. And if I remember right from the directions, let me just go peek right here. You need to count at least 50 of these spores, also called asci. So that's what it means by counting those. So how many black, black, tan, tan did you count? Please write that number here. I think the rest of this kind of walks you through it fairly straightforward. Make sure you get your total here. Again, this ratio is the black, black, tan, tan divided by the tan, tan, black, black. Just plug in your numbers. Make sure you take this times 100 to get your percent over here. Answer these questions down here. When it asks you if your distance was accurate, it says calculate your percent error. I've given you the equation for that. Please recognize that these values, let me go back there really quickly, are absolute values, so you will never have a negative number up here. And I want you to compare with what you got with what they got right here for their crossover frequency. And then make sure you state your conclusion. Let me go back here so we can see the rest of the lab. And then the final thing is the reflection. And another thing that a lot of students missed last year is it says make a personal statement about what you learned from doing both sections of the lab. A lot of people would just comment about the mitosis one or the meiosis one. A good comment not to make would be, it is really boring counting 100 cells in mitosis of the onion root tip cells. Give me something that's more significant than that, and you should be able to do well in this lab. Come see me if you have any questions, and I hope that helps.